parent cell divides in having the equal number of chromosomes. That is, since the chromosome number is equally distributed to the daughter cells, that is why mitosis is termed as divisional division. Yeah, समझ? तो हमने already discuss किया है. Now let's begin with the 3D animation of the mitosis. ठीक है? So let us begin. Here is the mitosis.
You will learn about mitosis. Mitosis is a process of cell division that makes possible the generation of body parts. To elaborate, mitosis is the separation of nuclear chromosomes into two identical daughter nuclei. It is divided into 4 key phases: prophase, metaphase, anaphase, and telophase.
Now let us begin one by one.
Phase called cytokinesis occurs after nuclear division. Before a dividing cell enters mitosis, it undergoes a period of growth and rest. This period is called interphase. In interphase, the genetic material of a cell increases.
These are centrosomes.
Prophase. During prophase, the chromatin condenses and becomes short and thick to form chromosomes. Each chromosome has 2 sister chromatids joined together at the centromere. The nucleolus disappears and the nucleus down. The centrosomes, each with a pair of centrioles, move apart to the opposite poles, forming a spindle between them. Metaphase. During metaphase, microtubules of the spindle interact with chromosomes. This causes the chromosomes to move and align along the middle of the cell or the equatorial plate.
It's an imaginary line.
Sister chromatids are held at the centromere, confirming the presence of protein structures called kinetochore at the surface. The kinetochore holds the chromosomes and attaches them to the spindle. Anaphase. During anaphase, the sister chromatids separate at the kinetochores, and the daughter chromosomes, along with the divided centromere, move towards the opposite poles as the microtubule shortens. The poles of the spindle also move apart and help to separate the chromosomes. As the chromosomes separate completely into 2 sets, one at each pole, anaphase is considered telophase. During telophase, the daughter chromosomes arrive at the pole and the chromatin starts to deploy. Also, the nuclei reform along with nucleoli. This triggers the reformation of the nuclear membrane. Cytokinesis. Cytokinesis is the partition of the cytoplasm. In animals, a cleavage furrow appears in the plasma membrane, which deepens and divides the cytoplasm into 2 daughter cells, each containing a nucleus. In plants, new cell wall formation is triggered at the center of the cell by the formation of a cell plate. The cell plate grows outwards till it meets the existing wall, resulting in 2 daughter cells. In this module, you have learned mitosis is the process of separation of nuclear chromosomes into 2 identical daughter nuclei.
Reduction division is the type of cell division that produces gametes. By gametes, we mean sex cells, such as sperm cells in males and egg cells in females. Meiosis is broken down into 2 stages of cell division, called meiosis I and meiosis II. Meiosis I has 4 phases: prophase I, metaphase I, anaphase I, and telophase I. And meiosis II also has 4 phases: prophase II, metaphase II, anaphase II, and telophase II. Let's look at what happens during meiosis I. Prophase I starts with a diploid cell. Its chromatin contains 2 uncoiled, spread out sets of chromosomes, 1 from each parent. After the DNA in the chromatin replicates, it condenses into the more familiar X-shaped chromosomes. The replicated DNA is the same in the identical sister chromatids of each chromosome. In a process called synapsis, each chromosome pairs up with and binds to its corresponding homologous chromosome, forming a tetrad. A tetrad is the group of 4 sister chromatids in paired homologous chromosomes. The chromosomes contain genetic information called genes. These genes were inherited from each parent. And different versions of the same gene on each chromosome are called alleles. In a process called crossing over, chromatids from each homologous chromosome exchange segments of alleles. Also called recombination, crossing over randomly happens on every chromosome, resulting in different gene combinations. This explains why every gamete is genetically different from every other gamete. Crossing over results in genetic variety in offspring. This is why children are different from their biological parents, as well as from their biological siblings. Continuing on with prophase 1, the nuclear membrane disappears. The centrioles move to opposite ends of the cell, and spindle fibers fan out from them. Next, in metaphase 1, the homologous chromosomes line up at the equator and attach to spindle fibers from opposite poles. During anaphase 1, spindle fibers separate the homologous chromosomes in each tetrad and pull them to opposite poles of the cell. The cell enters telophase 1 with 1 chromosome from each homologous pair at separate poles. However, each chromosome still consists of sister chromatids. Keep in mind that each chromosome's sister chromatids are no longer identical because of the allele exchange that happened during crossing over. Then, spindle fibers disappear and the nuclear membrane reforms around the chromosomes. Finally, cytokinesis occurs. Meiosis 1 ends with 2 genetically different haploid daughter cells. Each haploid cell contains only 1 set of chromosomes, consisting of paired sister chromatids. Both cells now enter the next stage, meiosis 2. However, unlike meiosis 1, DNA does not replicate before meiosis 2 begins. Once again, in prophase 2, the nuclear membrane disappears, and spindle fibers fan out from the 2 sets of paired centrioles. During metaphase 2, the chromosomes in each cell line up at the equator and attach to spindle fibers from both poles. During anaphase 2, the sister chromatids of each chromosome separate and move to opposite poles. Once the sister chromatids separate, they are called chromosomes. Finally, during telophase 2, the spindle fibers disappear and nuclear membranes reform. And cytokinesis occurs in both cells. Meiosis 2 ends with 4 genetically different haploid daughter cells, each containing only 1 set of chromosomes. Some key points to remember about meiosis. It begins with a diploid cell. Meiosis only produces gametes. Gametes are genetically different haploid cells, sperm cells in males and eggs in females. Meiosis has 2 stages of cell division, called meiosis 1 and meiosis 2. During meiosis 1, homologous chromosomes separate to produce 2 haploid cells, each containing chromosomes in the form of paired sister chromatids. In meiosis 2, the sister chromatids separate in both cells, becoming individual chromosomes. Cytokinesis of these cells produces 4 genetically different haploid gametes. And here are some key points to remember about prophase 1. The pairing of homologous chromosomes, called synapsis, occurs. Each pair of homologous chromosomes, consisting of 4 chromatids, is called a tetrad. During the process of crossing over, Chromosomes in homologous pairs exchange segments of alleles. Crossing over results in genetic differences in gametes. All gametes produced by meiosis are haploid.